This will be the one and only lecture for Chapter 10, which is on cell growth and division. The larger a cell becomes, the more demands the cell places on its DNA, and the more trouble the cell has moving enough nutrients and waste across the cell membrane. If we remember, cells diffuse through their membrane. So if they get too large, the amount of diffusion that can occur to meet the needs of the contents of the cell will not be able to be met by the overall surface area of the cell. By dividing the cell, it increases the surface area and decreases the volume and makes that surface area to volume ratio more usable for the cell. So as a cell grows in size, it does not usually make extra copy of its DNA, so it eventually grow too large to effectively function if the size was not kept in check. The larger the cell becomes, the more demand is put in the information in the cell. If demand grows too high, eventually it will not function. So there's not enough coding in the DNA for the cell to get too large. Also, like I said before, the exchange of materials into and out depends on the cell membrane. The surface area of the cell represents the area available for material exchange. As a cell grows, at a certain point it will divide, allowing for more surface area. The volume of a cell increases much more rapidly than the surface area, and cell division is the process where a parent cell divides into two daughter cells. So let's talk about that cell division right here. Chromosomes are made up of DNA, which carries the cell's coded genetic information. Before each division, each chromosome is replicated or copied. At the beginning of cell division, each chromosome consists of two identical sister chromatids, and the centromere is the area at the center of the sister chromatids that holds them together. So let's take a look at this right now. All right, we see the sister chromatids there separated. Basically what's going to happen is with the sister chromatids you have double the amount of genetic information, and they are going to divide and split those two sister chromatids apart, allowing for two identical daughter cells. So interphase is an in-between period of cell division where cell growth occurs. During the cell cycle, a cell grows, prepares for division, and divides to form two daughter cells, each of which then begins the cell cycle again. And mitosis is the division of the nucleus of the cell, and the nucleus in cell. And biologists divide into four phases: prophase, metaphase, anaphase, and telophase. So if we take a look right here, this is the cell cycle right here. G1 is growth, S phase is replication, the synthesis phase, and G2 phase is preparation for mitosis. And we see in the middle there, those are all interphase portions, so G1, S, and G2. And then the cell division phase, prophase, metaphase, anaphase, telophase, all are mitosis, and cytokinesis is the splitting of the cell. So interphase, the cell grows and replicates its genetic material. Prophase, the chromatin condenses into chromosomes and the nuclear membrane breaks down. Metaphase, the sister chromatids line up along the cell at a place called the metaphase plate. Anaphase, the sister chromatids are pulled apart into individual chromosomes. Telophase, the chromosomes gather at opposite ends of the cell and new nuclear membranes form. And cytokinesis, the cell divides into two identical daughter cells. So taking a look right here in the upper left-hand corner, we see interphase. Then prophase, we see the breakdown of the nuclear membrane. Then in metaphase, all the way to the right, we see the sister chromatids lined up along the center of the cell. And in anaphase, we see them being pulled apart. The sister chromatids separate. Telophase, we're forming two new nuclear membranes. So the cells will actually have two nuclei for a short period of time. And then, And then finally, cytokinesis is the division of the cell into two identical daughter cells. All right.